Hello friends, welcome to my channel Sam Biology Class. Today we will discuss about differential centrifugation. Before that, I request all of my friends, please subscribe my YouTube channel and also watch, like and share my videos with your friends. Differential centrifugation is a type of preparative centrifugation that separates particles of different size and density based upon differences in their sedimentation rate. This technique is also known as differential velocity centrifugation. Differential centrifugation utilizes multiple rounds of centrifugation at progressively higher speeds for the separation of various components based on their size and density. This allows for the separation of organelles and other cell components based on their sedimentation rate. Differential centrifugation can have several steps based on the level of separation desired. For example, it can begin with operating the centrifuge at low speed for the removal of undesired components. After the removal of undesired components, the sample is subjected to repeated centrifugation at progressively higher speed for the separation of various components present in the sample. In order to separate organelles, a tissue sample is first lysed to break the cell membranes to release the organelle and the cytosol. The lysate is then subjected to multiple rounds of centrifugation for the separation of various components based on their sedimentation rate. Differential centrifugation is also known as differential pelleting. This figure illustrates the principles of differential centrifugation. In this example, the tissue homogenate containing organelles, membrane vesicles and other structural fragments is subjected to multiple rounds of centrifugations by stepwise increase of centrifugal field. In first round, the tissue homogenate is subjected to centrifugation at a centrifugal field about 1000 G for 15 minutes. This results in the sedimentation of nuclei and other components at the bottom of the centrifuge tube as a pellet. After pelleting of nuclei and other components, the supernatant is transferred to another tube and is subjected to second round of centrifugation at a higher speed about 10,000 G for 15 minutes. The second round of centrifugation results in the sedimentation of mitochondria and lysosomes at the bottom of the centrifuge tube as a pellet. This is the pellet that contains mitochondria and lysosome. Because mitochondria and lysosomes are smaller than nuclei, in second round we applied strong centrifugal force than the first round, about 10,000 G for the sedimentation. So the second round of centrifugation results in the sedimentation of mitochondria and lysosomes. In the third round, the supernatant is transferred to another tube and is then centrifuged at a still higher speed, that is 1 lakh G for 1 to 2 hours. The third round of centrifugation results in the sedimentation of microsomes at the bottom of the centrifuge tubes. Pellets that are obtained from differential centrifugation are never pure. This is because they are mixed with contaminants. In order to minimize cross-contamination, each pellet is washed separately several times by resuspension in buffer and recentrifugation under the same condition. However, in some cases, particularly when we have a limiting amount of starting material, repeated washing may considerably reduce the yield of the final pellet fraction. In these cases, repeated washing is avoided. The final pellet fraction is then used for subsequent biochemical investigation. Differential centrifugation has several applications. Number one, separation of organelles and other cellular components. Number two, purification of biomolecules such as proteins and nucleic acids. Number three, isolation of extracellular vesicles including exosomes for research and diagnostic purposes. Number four, purification of nanoparticles, colloidal particles and viruses. Number five, study of cell biology phenomena such as organelle distribution, and additionally, differential centrifugation is also used to separate blood components such as red blood cells, white blood cells and platelets. Friends, this is all about differential centrifugation. For more updates, subscribe my YouTube channel, Sam Biology Class.